Very good evening, children. Can you all see me? Are you able to hear me? Shall we start the class? Okay, children, we'll start today's class. The topic is just continuous topic what we have learned last class. In today's class, we're going to talk about cytoplasm. The previous class, I have explained you about what is cell biology, and also we have learned about the structure of the cell, the various structure of the cell, and then we have learned about the plasma membrane. Today we are going to talk about cytoplasm. What is the cytoplasm? The cell has few components. The cell has the components. In that, the outer membrane is the plasma membrane, and next to the plasma membrane, cytoplasm is there. Cytoplasm, within the cytoplasm, the nucleus is suspended in it. So cytoplasm contains cell organelles and cell inclusions. So cytoplasm, it is a part that contains the cell organelles, as well as it contains cell inclusions. So you can see that the part that is present within the cell wall or cell membrane, we call it as cytoplasm. The cytoplasm contains these many organelles in it: vacuole, nucleolus, nuclear membrane, that's the nuclear structure, chloroplast, mitochondria, Golgi body, ribosome, endoplasmic reticulum, centrioles, centrosome, amyloplast, microtubules. All these are all structures that are present in the cytoplasm. This cytoplasm, it is a jelly-like substance which is present between the cell membrane and the nucleus. And this cytoplasm, it is a jelly-like substance because it mainly contains water. Apart from that, it contains the organic and inorganic components in it. In the cytoplasm, all the cell organelles are arranged, are suspended in the cytoplasm. Similarly, the nucleus is also present in the cytoplasm. Our nucleus are embedded in the cytoplasm. Apart from that, the cytoplasm also contains cell inclusions. The cell inclusions, it includes the storing materials by the cell organelles, secretions of the cell organelles, and excretion of the cells. And cell organelle that is present within the cytoplasm, it also contains various enzymes. These enzymes are responsible for various functions. For example, photosynthesis, respiration. For all these, lot of enzymes are involved in that. And those enzymes are present in the cytoplasm, present in the cell organelles that are present in the cytoplasm. So cytoplasm is known as the site of chemical reactions within a cell, because most of the cell organelles are present in cytoplasm. That is the reason why the cell cytoplasm is known as the region of chemical reactions or biochemical reactions within the cells. The cell inclusions. The cell has reserve food. For example, the starch grains in the potato tuber. The potato, it is called as tuber. It stores the food material. Similarly, the glycogen granules in liver and muscle cells, because glycogen is a complex form of glucose in animals. Protein granules. If you see the monocot seed, there will be one layer that covers the endosperm, that is called allulane layer. Allurane layer. This allurane layer, it contains protein granules and oil droplets in the case of adipocytes, which is present beneath the skin. The endosperm cells of castor, coconut, and cotyledon of groundnut seeds are all oil seeds. It contains oil droplets that are all reserve food materials. Gases. The cell cytoplasm also contains oxygen, carbon dioxide, and nitrogen. Apart from that, it also contains inorganic crystals, silica. Silica in the epidermal cells of grasses. In the upper epidermis of the grass, you can see some cells called the bully cells. Those bully cells, it contains silica. Silica means sand particles. And calcium carbonate crystals in the epidermis of Mimodica and calcium oxalate crystals in the ecornea. These are all the inorganic crystals that is present there. Apart from that, the cytoplasmic inclusion, it also includes the excretory product. There are certain excretory products that are produced by the cells which has an impact, very good impact, like help in formation of medicines. It become an ingredient of certain medicines. Mucus, tannins, resins, gums, alkaloids, and latex, the milk-like substances that comes out from certain plants. These are all the excretory products. The latex, it will be there in banyan, calotropis, oleander, and papaya, and also you can Havia bracenalis. This is a rubber tree which yields rubber. Gums, the gums in the case of moringa plant, moringa, and in pine forest and casuarina, you can see that some gum-like substances excrete out from the stem. That is a product of cell wall. It's a product of cell wall. Alkaloids. Quinine, this is an alkaloid which is extracted from the bark of Chinchona officialis plant. Atropine, Atrophobelozona. Nicotine, Nicotinamide tabacum. Morphine, Papaver somniferum. Colchicine, Colchicine autumnale. These are all plants that excrete the alkaloids. The colchicine, it is used to induce the doubling in chromosomes. Morphine, painkiller. Nicotine, atropine, quinine. These are all medicines which are used to treat various diseases. Essential oils. The essential oils, it includes the lavender, some aromatic compounds, lavender, rosemary oil, menthol. Menthol is used to make a lot of medicinal products, painkillers, and eucalyptus oil. These are all extractions from the cytoplasm. Nectar, which is from the flower, a sugary substances that is present, that is produced by the flower, which contains glucose, fructose, and sucrose. That is called nectar. That is also excretory product. And pigments, chromatophores. In the case of fishes, the skin of fishes, it has various colors. Similarly, the amphibians and reptiles. Melanophores, which are called brown pigments. Lithophores, red, yellow, and orange pigment. These are all pigments that are excreted which are present in the cell cytoplasm. Next, we are going to talk about the cell organelles that are present in the cytoplasm. The cell organelles which are present in the cytoplasm, they are all membrane bounded organelles, or there are certain membranes, certain organelles without membrane is there. Some organelles, they have two membranes, double membrane, and some are single membranes. And cytoplasms are small microscopic organs which are present within the cytoplasm. There are three categories: without membrane, single membrane bounded organelles, and double membrane bounded organelles. For example, the ribosome, which are known as the protein factory, they are all without cell membrane. They are present in both prokaryotic organism and eukaryotic organism, primitive organism as well as the true organisms. In both the organisms, the ribosomes are present. They are mainly involved in protein synthesis. And there are single membrane bounded organelles. Vacuole contains chloroplast, lysosome, Golgi apparatus, endoplasmic reticulum. They are present only in eukaryotic cells because the membrane bounded organelles, these membrane bounded organelles, understand children, the membrane bounded organelles are absent in the case of prokaryotic cells. They are present only in eukaryotic cells. And double membrane bounded organelles like mitochondria, we can see that the outer membrane and inner membrane, it forms the cisternae, the inner foldings, and chloroplast, double membrane again. These are all present in eukaryotic cells. And some of the cell organelles, we are going to talk about that. One is the endoplasmic reticulum. What is this endoplasmic reticulum? What is the function of endoplasmic reticulum? These are all typical structure that is present in the cytoplasm, which is known as the skeletal of the cells. They give a definite structure to the cells. And these endoplasmic reticulum, shortly we call as ER. ER, it is the interconnected membrane bounded canals which connects the outer membrane, that is the cell membrane, with the nucleus. So right from the cell membrane till the nucleus, you can see lot of channels like this, canals like this. These canals are known as the endoplasmic reticulum. This was first identified by Porter and Kalman in the year 1952. And this endoplasmic reticulum, it exists in three different forms. One is the cisternae. So this structure like this is called cisternae, the foldings. They are in the form of vesicles, round structures. In the form of tubes, they are called tubules. These are the three different types are there. Apart from that, these endoplasmic reticulum, there are two different types of ER is there. One is RER and SER. What is RER? If the ribosomes are present in the endoplasmic reticulum, it is called RER. If ribosomes are absent, it is called SER, smooth endoplasmic reticulum. And RER, it is found throughout the cell. Since it contains the ribosomes, the ER are involved mainly in manufacturing or synthesis of protein, because ribosomes are known as protein factories. So ribosomes along with RER, it does the process of producing proteins. Whereas the SER involved, it is a storage organelle. It is involved in synthesis of steroids and lipids, and also responsible for detoxifying the cells, a removal of toxins. That is also done by smooth endoplasmic reticulum. So RER, SER, you can able to differentiate, right? RER means it contains ribosomes in it. And SER, ribosomes are absent. And what are the main functions of this endoplasmic reticulum? As I mentioned there, the ER, it attaches from the outer membrane, the plasma membrane, it connects the nucleus. So all transportation of molecules within the cell are transported, or transportation from the cell to the out. Everything is done with the help of the transporting system, which is known as the endoplasmic reticulum. And also the endoplasmic reticulum, it is made up of cisternaes. The foldings, it will be there like that. It has lot of foldings, it will be there. So that provides lot of surface area for various metabolic activities. For example, protein synthesis of lipid, synthesis of steroid, everything is done within the space provided by the endoplasmic reticulum. So that is about the main functions of ER. The next organelle is called ribosomes. Ribosomes are organelle without the membrane, without any membrane. This was first observed by George Palade in the year 1953. And the electronic microscope view of cell organelles, they have identified this ribosomes consist of two units, one large subunit and one small subunit. And also for attachment of the the large subunit and small subunit, the Mg2 plus ions play an important role. And another important thing about ribosomes, the biogenesis. What is biogenesis? Formation of ribosomes are de novo formation. That means auto replications. The ribosomes are formed from the ribosomes. Or it can be a nucleolar origin. Nucleolar origin, because the nucleolus are also involved in protein synthesis. And also ribosomes are formed from the nucleolus. So they can form by themselves, auto-replication, that is self-replication process, or nucleolar origins. And both the largest subunit and smaller subunit, both are the site for protein synthesis. The protein synthesis, that is the formation of protein, that process is called translation process. Translation. That translation process, it occurs mainly in the ribosomes. So ribosomes, can you see this process? This is the process of formation of the protein. This is the structure, what you are seeing it here, the largest subunit and smallest subunit. Can you see it here? This is the smallest subunit, the largest subunit, and according to the code that is there in the mRNA, this is called mRNA. By a process called transcription, mRNA is formed. By a process called translation, proteins are formed. Transcription, formation of mRNA from DNA. Make a note, children. Translation means formation of mRNA from DNA. That's called transcription. Translation means formation of proteins according to the code given in the mRNA. So here you can see that lot of amino acid joined here. All those amino acid joined here, it is because of the code that is present in the mRNA. இங்க இதுல என்ன இருக்கும்னா கோட் இருக்கும். mRNA ல கோட் இருக்கு. அந்த கோடுக்கு ஏத்த மாதிரி தான் என்ன ஆகும்னா அமினோ ஆசிட் ஃபார்ம் ஆகும். இந்த அமினோ ஆசிட்லாம் ஒண்ணா சேர்ந்து புரோட்டீன் ஃபார்ம் ஆகும். So ribosomes, it consists of RNA and protein. 40% it contains protein, 60% it contains RNA. And this RNA, the whole RNA, it is involved in the process of protein synthesis. So ribosomes are also known as polysomes, or polyribosomes, or ergosomes. The functions of these polysomes, it is mainly production of protein from the amino acids. So many amino acids, they are linked by peptide bonds. They are linked by peptide bonds and they form the proteins. And here the linking together, it is done by Mg2 plus ions. And main functions of ribosomes, as I mentioned there, they are involved in the process of translation, that is synthesis of protein. And also proteins are the main structures that helps in formation of enzymes, formation of hormones, formation of various body structures. So directly the synthesis of ribosomes, that is the protein synthesis, it ensures the survival of the life. That is about the functions of ribosomes. And the types of ribosomes, there are two different types of ribosomes are there. The ribosomes, it varies in prokaryotic ribosomes and eukaryotic ribosomes. This is the one of the organelle that is present in both prokaryotic as well as eukaryotic cells. In the eukaryotic cells, the ribosome, it is 80S. S is the unit that is called Svedberg unit, that is called S. This S, it means Svedberg unit. This is named after Theodor Svedberg, who is a Nobel laureate, and this unit, it explains about the particle size dependent on the speed at which the sedimentation rate takes place during centrifugation. According to the sedimentation rate, When you centrifuge something, centrifuge means rotating something at high speed. What will happen after that? Sedimentation process takes place. That sedimentation of the particle depends on the size. Similarly, this 80s, it means the Svedberg unit, it represents the particle size. 80s, it contains 2 units. In the 80s type, 2 units are there. One is the 40s and 60s. The small unit is called 40s unit, the large unit 60s unit. And you can see that different types of RNA are present. 5s RNA, 5.8s RNA, 28s RNA and 18s RNA. These are all different types of RNA or different units of RNA that is present in eukaryotic ribosomes. Next is the prokaryotic ribosomes. The prokaryotic ribosomes, it is present in prokaryotic organisms like blue-green algae, bacteria, and in mitochondria it will be there. In chloroplast of many algae and higher plants, we can see that 70s type of ribosomes are present. This 70s type of ribosomes, it contains 5s RNA similar to 80s, 23s RNA and 16s RNA. And the unit, you can see that one is the largest subunit is 50s unit and smallest subunit is 30s unit. This is about the types of ribosomes. Next we are going to talk about lysosomes. Lysosomes. Okay, so before explaining the lysosomes, I'll just clarify your doubts and then I'll move on to that. And Madhe Krishna, in reverse transcription, the mRNA gets converted into DNA. Yes, that is reverse transcription. I'll just show you one picture that is called the central dogma. I'll just show it to you. You can understand from that. You can also write that. The central dogma of biology, where DNA forms the DNA. DNA, it forms the DNA. That is called, just 1 minute children, I'll just show it to you. This is the central dogma. Are you able to see that, children? Central dogma. Can you see it here? So you can see this. The DNA, it undergo a process called replication. That is called DNA replication to form DNA. So one DNA, it becomes 2 DNA. That is called DNA replication process. And from the DNA, it is a double stranded structure. Double stranded structure. From that, a single stranded RNA is formed. This RNA is called mRNA. It's called messenger RNA. Messenger RNA by a process called transcription. And this mRNA or this mRNA, it can become DNA. That process is called reverse transcription. Reverse transcription. And mRNA, it undergo a process called translation and forms protein. This is called central dogma of biology. Sometimes they'll ask in board exam or school exam, they'll ask you to write about central dogma of biology, where you have to write DNA undergo a process called replication and forms DNA. DNA undergo a process called transcription and forms mRNA. mRNA undergo a process called translation and forms protein. This is the central dogma. And the next question, Vashini, is the 80s ribosomes bigger than 70s ribosomes? It's not whether it's bigger or smaller, ma, and it is mainly the sedimentation rate. That is called sedimentation rate. The size does not make the difference. Usually what will happen in eukaryotic cells only, the ribosomes are bigger in size when compared to the prokaryotic cells. And also sometimes we'll get confused with sir, here it is given as 70s unit, but you can see that It is 70s means 50 plus 30 it become 80. Some people they used to ask a lot of doubts like that, but it is not so. Here they are actually calculating the units. These are the DNA, that is the RNA that is present inside. That calculation only it is 70s. Sometimes it vary in organisms. That's why this sometimes here they'll have more and here it is be less. And similarly here also. In 80s type it will become 100s unit. But the structure is eukaryotic is 80s. That is a standard structure. It might vary. That's why it is given as different forms. And Madesh, next question. The coronavirus undergoes reverse transcription, right? Yes. Not only corona, most of the viral diseases, they undergo a process called reverse transcription. Reverse transcription. Because, for example, the AIDS, the HIV virus also, the structure consists of, it is called retrovirus, and the structure consists of 2 RNA molecule, and along with that reverse transcriptase, there is an enzyme called reverse transcriptase is there. That enzyme, it converts the 2 single stranded RNA into mRNA, into DNA. Yes, you are right. Okay children, so before going to the next topic, lysosomes, we'll go for a short break for 5 minutes and then we'll come back. Okay children, just go for a break, drink some water and come back. I'll explain you about the lysosomes after this class. What about others? Kadiravan, Vashini, you have asked the doubt. Deepak Ishwar, Balakrishna, Subarna Lakshmi, Kishore, Deepak, and Kadiravan, any doubt? Clear? Okay, very good. Thank you. So Charvi is absent, right? I don't know why. Clear? Okay. Thank you, ma. அப்பப்போ இந்த மாதிரி கொஞ்சம் மெசேஜ் போடுங்க. அப்பதான் நம்மளுக்கு என்னன்னா whether you understood or not, I need to know it, right? புரிஞ்சு தானே படிக்கணும், அதனால தான் கேட்குறேன். உங்களுக்கு எதனா புரியலன்னா என்கிட்ட சொல்லுங்க, நான் சொல்லிட்டேன். ஓகேமா? Are you all clear? Okay, go for a break. We'll come back after 5 minutes. Shall we continue? Are you all there? Are you all there? Shall we continue? Okay. In the WhatsApp, I have sent the answer key for the test what you have written, the transcription, the transportation in plans. Do you remember that? Okay. I have sent it there. So kindly check the answers and send your marks to me personally. Okay. Thank you. So we'll start the class in 2 minutes. One more person is going to join now. So I'm just waiting for that boy to join. Just give me 2 minutes time. We'll start the class. Okay. Shall you tell me how we are all performing in your school exams? Shall I discuss about that for a few minutes? How we are all performing in school exams? Welcome, Madesh. This is Paul sir. I am your biology teacher. So this is our class. I will teach here by using this particular software only. Okay. So if you have any doubt, Madesh, you can type it here. You can type it there so that I will respond to you during the break time, not during the class hours. During the break time, I will do it. Okay. And yes, welcome to the class. We'll continue it. Okay, other children, what you can do it is after the class, that is after 8 o'clock, you can do the correction and send it to me by 8:30, the same format what we have sent it last class. Okay. So just for information about Madesh, we are actually discussing about the cell structure and functions, and we have completed, actually we are doing cell organelles here. We have done with some topics there. That link I'll send it to you later where you can go through it. Okay. So this is lysosomes. Next class. Okay, I'll just check it and send it to you again. Again I'll do it. Okay, no problem. So answer key, that last line it was not seen properly. I'll type it. I will send that later to you. Okay, children. Done. Okay. So we'll start the class. The next topic, it is about the lysosomes. Lysosomes. The term lyso means digesting. Soma means body. So what is the role of this lysosomes? This lysosomes was first discovered by Christian D. Duke in the year 1953. This lysosomes, can you all see that? This is the structure of lysosomes, where it contains a particular enzyme called hydrolytic enzyme. Hydrolytic enzyme. This hydrolytic enzymes are mainly used in digestion process. You might have seen about the phagosomes or phagocytosis process. In the phagocytosis process, the lysosome joined with the phagosome and they form phagolysosomes, and then only digestion process takes place. The digestion or killing of the foreign bodies, it is done by the lysosomes. That's why the lysosomes are known as suicidal bags. And the membrane, the lysosomes, the membrane around the lysosomes. Can you see it here? The single membrane around the lysosomes. The enzyme, the hydrolytic enzymes will not digest the cell itself. So the cell membrane are protected towards the hydrolytic enzymes. But this hydrolytic enzymes, it kills the other cell structures, or it can kill the other foreign bodies. It can digest them, but it won't digest the cell itself. That is the functions of that. So where these the lysosomes are present, these lysosomes are present in WBC, where it helps in killing the pathogens. That is called phagocytosis process. And osteoclasts, the bone cells, and also the other cells also, the lysosomes are present. And lysosomes, they are absent in plant cell, but in the case of neurospora, as well as the tip of the maize, yeast, as well as the cotton plants, the pea plants, some lysosomes are present, but they will be in very less in number. And here you can see these are list of lysosomal enzymes that is present in the lysosomes, which are called as the hydrolytic enzymes. Phosphate esters are present. They are called as acid phosphatase. Nucleases, with digest the nucleus, right? DNAs and RNAs are present. Protein digesting enzymes. Collagenase, which digest the collagen. And cathepsins A and B. Peptidases, to break the bond between the protein molecules. And proteases. These are all protein digesting enzymes present within the lysosomes. Similarly, they also have carbohydrate digesting enzymes, which digest the complex carbohydrate like starch and glycogen into glucose, fructose, galactose and mannose. Beta galactosidase, hexosaminidase A, alpha mannidase, fucodases, silidases and glucuronidase. These are all some of the enzymes that is digesting the carbohydrate digesting enzyme that is present within the lysosome. And also they have lipid digesting enzymes. Acid lipase, phospholipase, phosphatidic acid phosphatase, esterases, sphygmomyelinase. These are all some of the enzymes that are present in lysosomes. All these enzymes, they are involved in digesting the cellular structures, the biomolecules that are present in the cells of other organisms like foreign bodies. This is the role of lysosomes. Next we are going to talk about the functions of lysosomes. The functions of lysosomes are involved in intracellular digestion. Intracellular digestion. What is this intracellular digestion? The cells, they can, intra means within, the digestion that occurs within the cell, that is called as intracellular digestion. Intracellular digestion means digestion occurs within the cells. What they will do, these lysosomes, it attaches with other cells and form phagolysosomes and brings the cell inside and kills them. That's why it is known as the it involved in cellular digestions. Next is it is involved in autophagy. It involved in autophagy. What is this autophagy is all about? Autophagy means it is a natural regulated mechanism of the cell that removes the unnecessary and dysfunctional components out of the cells. In one way, you can tell that is degradation of the cell particles. cellular components, or recycling of cellular components, or excretion of cellular components, that is done by the lysosomes. Similarly, membrane resealing is also done by lysosomes. And pathogen entry, it prevents the entry of pathogens. During phagocytosis process, it kills the pathogen with the help of the protein digesting enzyme, carbohydrate, lipid digesting enzyme. With the help of that, it kills the pathogens. And also, these lysosomes are present in osteoclast cells, that are bone forming cells, where it helps in bone matrix reabsorption. And also lysosomes, with the help of lysosome, the identification of antigen presentation can be identified. What is antigen presentation? When any pathogen enters inside, it releases the toxin substances within the body, that is called antigen, and those antigen can be identified with the help of lysosomes. And also lysosomes are involved in cholesterol transport, and they can remove carcinogens. What is carcinogens? What is carcinogens? The cancer causing agents. The cancer causing agents. And also breaking down of aging and dead cells, that is again called as autophagy, that is also done by lysosomes. Okay children, now we can copy this slide. I'll give you 2 minutes time. I'll be in online. So copy this slide, take a photograph and send it to me in WhatsApp. Do it children. Write the functions of lysosomes and take a photograph and send it to me immediately. All of you do it. Take a note, copy the functions of lysosomes, all these functions, take a photograph. Yes, Pashini, you asked a very good question. If lysosomes can remove carcinogens, then why are cancerous deadly? For this question, the answer is, the cancer, it is not going to attack only one cell. The cancer, it mainly, once it gains entry, if it is going to be because of the oncogenic viruses, then it can be somehow, it can be controlled by pathogens, because cancers are mainly not only because of biological agents, there are chemical and physical agents also involved in causing cancer. In those cases, the lysosomes cannot act immediately in all cells, because already the lysosomes, it has lot of functions to do in the cells. For example, I have listed out so many functions here. All these functions are carried out by the lysosomes. Along with that, if they are able to remove the carcinogen, that is fine. But immediately the lysosome, the primary functions of lysosome is not to remove the carcinogens. It is mainly intracellular digestion and controls the pathogen entry. That is the reason why the cancerous cells, we know that the different stages of cancers are there. In that, through the blood, the cancer can be transferred to different parts of the body. That is called metastasis stage. In those stages, the lysosomes will not function properly. That is one of the reasons why the cancer will become a deadly disease. Have you understood, Vashini? Suparna Lakshmi, you want to tell something? You can tell. No problem. You have doubt, child? You can tell me. Madesh, Madhav Krishna, do you have any doubt? I'm just waiting for your response, children. Thank you, children. Okay, just do that. Write the functions. Post it now. Sure, Kajiravan Balakrishnan, no doubt. Very good. Suparna Lakshmi, you want to tell something, ma? You want to tell something? Please tell now. So I have received one from Madesh, Madhe Krishna. Others? Balakrishnan, yes, I got it. Suparna Lakshmi has sent it. Very good. Children, hereafter I want to make it mandatory in my class that you have to take notes and upload the class notes immediately after the class. It become mandatory. Are you all clear, children? Are you all clear, children? Just take the summary points alone. Don't write all the points whatever I am telling. Just keynote summary points. You write it in a A4 sheet and after the class immediately upload it to me. You can send it to me and attach the same in your textbook or whatever note you have, because that is the summary points will help you to revise the topic. Got it? All of you got it? I want all of you to follow that from next class. Even if you are following from this class, I am very happy. Vashini has sent it. Waiting for others. Madesh, you have the group number. Kishore Balamurugan has sent it. Usually Kajiravan will be very fast. I don't know what happened to Kajiravan today. Deepak also sent it. Very good, Deepak. Okay, children, we'll continue the topic. Thank you very much. Thank you for your immediate response. Next is about Golgi body. The Golgi body is also called as dictyosomes. Dictyosomes. And this Golgi body, it was first observed by Camillo Golgi in the year 1898. And this is a membrane bounded organelle, and you can see that this membrane, this particular organelle, it exists in different forms. Can you see that inner foldings like structure? This is called cisternae forms. They can also be in tubular forms like a tubes, and they can be in vesicles. Can you see it here? These are all vesicles, and vacuoles like large size also it can be there. So it can exist in 4 different forms, it will be there. And in the case of plant cells, this Golgi body, there are like observe as small vesicles. Those vesicles are known as dictyosomes. Dictyosomes. So they can exist in 4 forms: cisternae, tubules, vesicles, and Golgi vacuoles. Next, we are going to talk about the functions of Golgi complex. The functions of Golgi complex, it mainly involved in production of glycoproteins and glycolipids. Do you remember what is the function? Where exactly you have seen glycoproteins and glycolipids? Can anyone tell? Where exactly you have observed these two terms, glycoproteins and glycolipids, where they are formed, glycoproteins and glycolipids? Can anyone tell where they are formed? Fluid mosaic model. Very good, ma. What is that? Actually, fluid cell membrane. Very good, Vashini. Very good, excellent. So, in the glycoproteins, in the walls of the biomembrane, that is the cell membrane, you can see that the cell membrane is made up of glycoproteins, glycolipids, phospholipids, bilipid layer, all those things it is there. So, here now you can understand from where these glycoproteins and glycolipids are formed. And transportation and storing of lipids, because the main functions of Golgi complex, it helps in transportation of materials. Like a courier it will act. And also it is involved in formation of lysosomes. The lysosomes are mainly formed from Golgi complex. And other functions, it also involved in formation of digestive enzymes which are present within the lysosomes. You have learnt about the carbohydrate digesting enzyme, protein digesting enzyme, lipid digesting enzymes, DNAs. All these enzymes are also formed from Golgi complex. And also these are involved in cell plate and cell wall formation. What is cell plate and cell wall formation? During the cell division process, do you remember that the cell divides into two, and there are two methods of the formation of daughter cells. Daughter cells separate in two methods. In that, the cell plate method and cell wall formation, these two are mainly formed with the help of Golgi complex. And also the secretion of carbohydrates for the formation of cell wall. Carbohydrate formation of cell wall, right? That is also done by Golgi complex. And also in the case of animals, the cutin. You can see that the insects, the outer skeleton, it is made up of cutin. It's called exoskeleton. It is made up of cutin. So that cutin is also formed from Golgi complex. And apart from that, Golgi complex, it contains zymogen granules. These zymogen granules, they are all the proenzymes. These zymogen granules only become different type of enzymes. It is a precursor for all enzymes. From this zymogen granules, so the different kinds of enzymes are formed. And also the ultimate function of Golgi complex, transporting. It helps in transporting the material, modifying the material, packing the proteins, packing the lipid, transporting them to the target destinations. That is the role of the Golgi complex. Next, we are going to talk about mitochondria. The mitochondria, it is known as, it is a autonomous organelle. Autonomous organelle. Why it is called autonomous organelle? I'll tell you why it is called autonomous organelle later. The mitochondria, it is a membrane bounded organelle. It has two membranes, the outer membrane and inner membrane. This was first observed by Kolliker in 1880 and Altman in the year 1894, and Altman named it as bioplast. And the term mitochondria, it was given by Benda. Sometimes, usually what will happen, lower classes we might have learnt about who discovered mitochondria, will tell Benda. But this was first observed by Kolliker, later by Altman, and then only Benda. And mitochondria, as I mentioned, there are two membranes, the outer membrane, inner membrane. The inner membrane, it has inner foldings. Lot of inner foldings are there. These inner foldings are mainly to increase the surface area for respiration. And also the outer membrane, can you see this is the outer membrane? The outer membrane, it has lot of protein channels are there. That's called porins. These porins, they are all involved in movement of molecules. The small molecules, it can enter into the mitochondria with help of the small pores that are present in the outer membrane of mitochondria, that is known as porins. Similarly, the inner membrane, can you see the inner membrane has lot of inner foldings? It will be there. These are all inner foldings. These inner foldings are called as cistae. So this inner membrane, it has lot of infoldings. That inner foldings are known as cistae. That cistae contains enzymes for electron transport system. And the inner chamber, this is the inner chamber. The inner chamber of mitochondria, they are filled with proteinaceous material. It contains lot of proteins or enzymes are present there. So this is known as the matrix, the base. And within the inner membrane, can you all see it here? The structure within the membrane, inner membrane, you can see lot of stalked particles like this. Those particles are known as F1 particles. F1 particles. These F1 particles are also known as Fernandez-Moran particles, are elementary particles. They are also called oxysomes. The structure of this, it has a base and a stalk called pedicel and a head. This is a typical structure of the F1 particles. This F1 particles are oxysomes. They are mainly involved in oxidative phosphorylation. What is phosphorylation? Formation of ATP. The formation of ATP is known as phosphorylation, because this structure, it contains an enzyme called ATP synthetase. This ATP synthetase enzyme, it is involved in formation of ATP. So you know that the ATP, the energy molecules, are mainly formed in the mitochondria. The reason why the mitochondria, it is known as the powerhouse of the cell. So that formation of ATP, the ATP synthetase enzyme is very important. That is present in the F1 particles, which is present in the inner foldings of inner membrane. And this is the typical structure of mitochondria. You can see that in the outer membrane and inner membrane is all, it is the inner mitochondrial membrane. Lot of inner foldings are there. So mitochondria contains 73% it contains protein, 25 to 30% it contains lipid, 5 to 7% contain RNA and DNA. Some traces of DNA it will be there, and it has respiratory enzymes. 60 different types of respiratory enzymes are present in mitochondrial matrix. Here, can you see it here? These are all the matrix regions, mitochondrial matrix. And mitochondria, it is known as semi-autonomous organelle. As I mentioned that it is called semi-autonomous organelle. Why? Because it has its own DNA. Usually if you take a cell, the cell has a nucleus, that nucleus contains DNA, whereas the mitochondria being an organelle, it has its own DNA. That's why it is called semi-autonomous organelle. And also it has ribosomes, 70S type of ribosomes, which is usually seen in prokaryotic organisms. They can even multiply themselves by a process called fission and also replicates. So one mitochondria become two mitochondria. They can self-replicate. And this is the site for respiration. It is a respiratory organelle. So this respiratory process, that is the formation of ATP with the help of glucose. The glucose molecule is oxidized. The oxygen is added to the glucose molecule. That's called oxidation of food to release energy. That process is called respiration. Can you all see it here? This oxygen, it combines with glucose molecule. That's called oxidation of food to produce energy. During this process, carbon dioxide and water are produced. So what is the ultimate aim of respiration? That is formation of ATP molecule. Formation of ATP. That is the ultimate aim of the respiration process. That is the reason why the mitochondria, it is known as the powerhouse of the cells. Why it is called powerhouse? Because it produces what? ATP. That ATP is the one which gives energy for all physiological process. That is the chemical energy. It provides chemical energy for various physiological processes. That's why the mitochondria, it is known as the powerhouse of the cell, and ATP is known as the energy currency of the cell. Next, we are going to talk about peroxisomes. The peroxisome is an organelle which was identified by Christian de Duve in the year 1967. This is also a spherical structure like vacuoles, or spherical structure like lysosomes, and you can see that they have single membrane bounded organelle. Even like plasma membrane, these peroxisomes are also made up of bilipid layer. A lipid layer is there. These peroxisomes are involved in photorespiration, and also the glycolate metabolism, it occurs in peroxisomes. Two functions, one is photorespiration and glycolate metabolism. Peroxisomes, where exactly these cells are present, these organelles are present, they are present in leaf cells, in the liver, kidney of mammals, and also they can see in protozoans and yeast. That is about the peroxisome. The functions, peroxisomes are the one which helps in formation of water and oxygen. So this peroxisome it contains an oxidative enzyme that is known as catalase. This catalase, it breaks the hydrogen peroxide that is produced in the cells into water and oxygen. That is the main function of peroxide. It protects the cell from the production of hydrogen peroxide. Once hydrogen peroxide is formed, the peroxisome immediately releases the oxidative enzyme called catalase. This catalase, it breaks the hydrogen peroxide into 2 molecules of water and 1 molecule of oxygen. This is the function of peroxisome. Next, we are going to talk about centrioles. So we'll take 2 minutes break. Meanwhile, if you have a doubt, I'll clarify a doubt. 2 minutes break, and after that I'll come and join with you. You also go and drink some water and come back. After 2 minutes, I'll join again. Okay, children? Thank you very much. Welcome back, children. So before explaining about the centrioles, I'll just go through your doubts. I'll clarify your doubts. Vashini, if mitochondria has its own DNA, can it live on its own without the plasma membrane? See, mitochondria, it is not considered as separately as a cell. It is an organelle. A heart cannot be, it will not function separately as an organ. You got it? A kidney, even though it is an organ, it cannot be an individual. It is not an organism. The organism means it should contain different types of organs to perform different types of functions in this. This is an semi-autonomous organelle. Not only this, even the chloroplast are semi-autonomous organelles. They cannot live separately as a cell. You got it, child? Thank you very much. Kadiravan, why hydrogen peroxide should be broken down into water and oxygen? Because, very simple thing, hydrogen peroxide, it is an highly dangerous because it is a strong oxidizer and it causes lot of diseases. It causes corrosive to the eyes, irritation to the eyes. It causes skin infections, and also it can damage your eyes. It can damage the respiratory system. It can damage the respiratory system. Suppose when you are inhaling that high concentration of hydrogen peroxide, when you take hydrogen peroxide more, it can cause ulcer right from the respiratory tract. It can cause lot of ulcers, and also in the gut region, mouth, throat, stomach region, everywhere it can cause ulcers and it causes burns. Higher accumulation of hydrogen peroxide in our body, it can even lead to death. Got it, child? Okay. So next question, Vashini. Water is already produced from peroxide in peroxisomes. Why do plants cells require water from environment? Very simple thing. The water molecules that are formed from the catalysis of hydrogen peroxide, it is very, very less. And you know that the transpirational pull, it takes lot of water. 98% of water is transpired out. Only 2% the plant will use it. And very little percentage it was produced by the peroxisomes. Got it, child? Okay, next we'll move on to, are you clear, Vashini? Okay, man. Others, do you have any doubt? Madhesh, Madhe Krishna, Balakrishna, Subarna Lakshmi, Deepak, Kishore, Balakrishna, doubt irukama? Okay, no doubt. Very good. We'll move on to the next topic. The next topic is centrioles. Centriole, this is an organelle that is present exclusively in the animal cells. In the animal cells. The structure, pathina, the centrioles, the pattern It is 9 plus 0 pattern, triplets. இந்த மாதிரி தான் இருக்கும் structure. Can you see that? The structure of centriole, how it is arranged. It is a 9 triplet made up of peripheral fibers. This is called tubulin. Tubulin proteins. The center part is there, right? The center part, this is called hub. These are all tubules. The tubules are connected to the linker. This is called AC linker. And with that you can see that this is the pinhead. This is the AC linker which links all the 9 peripheral fibers. And you can see that this is the pinhead, and the central hub, this is the central hub. These are tubulin spokes, radial spokes, and radial spokes, it will end up in a pinhead structure, and these are all the microtubules. Microtubules are there, and this is a typical structure of centrioles. The centrioles are form the basal form of cilia, which helps in organ of attachment, flagella, which helps in locomotion, and also during cell division, the spindle fiber formation. Can you all see that? This is the spindle fiber formation, which helps the chromosomes to arrange at equatorial plate, and also separation of chromosomes. The spindle fibers forms an important role. So these spindle fiber, it is also formed from centrioles. Next we are going to talk about plastids. Plastids are the different types of plastids are there. I'll just explain that and then explain about the structure of plastid, the structure of the chloroplast. I've already explained you in the previous topic. So even if you want, I'll just explain that also again. So these plastids, it was term was first used by Schimper in 1883. Like mitochondria, this is also semi-autonomous organelle because it has its own DNA. And also the plastids, mainly the chloroplast, it resembles the character of cyanobacteria. So according to the evolution, these chloroplasts might have been evolved from cyanobacteria, because the cyanobacteria as well as the chloroplast, all of them, they have similar proteins, lipids and carbohydrates. Different types of plastids are observed in organisms. One is called etioplast, chromoplast, chloroplast, leucoplast, amyloplast, ileoplast and proteinoplast. The proteinoplast, it is, you can see in site of enzyme activity or protein activities. Ileoplast, they are involved in storage of lipids and oils. Amyloplasts, they are involved in storage of sugars and starch. Starch is a complex form of glucose. Leucoplast, synthesis of fatty acid and amino acids. Chloroplast, it involved in lipid synthesis as well as proteins, the photosynthesis process. Chromoplast, pigment synthesis and storage. Etioplast, they are involved in crystalline prolamellar bodies and is a precursor of chlorophyll, because from etioplast only the chlorophyll molecules are formed. This is about the types of plastids. In that, the main plastids are the chloroplast. Chloroplast is again a double membrane organelle. The double membrane, it is evidence for endosymbiosis. That is an independent origin. That's what I told you. It might be evolved from cyanobacteria. And just assume that the room in which you are sitting, it is a chloroplast. You have one wall, right? Think that above that one more wall is there. Like that, 2 layers, it will be there in plastids. And the floor is called stroma. The stroma, it has lot of enzymes that are involved in the process of photosynthesis. Similarly, the stroma, it contains a sac-like structure called thylakoid. This thylakoid, it has the electron transport system as well as the photophosphorylation, formation of ATP, it occurs in thylakoids. And this thylakoid, it is a single structure. The single sac-like structure is called thylakoid, and many thylakoids, they are arranged one over the other. They are arranged one over the other, and they form a pile of coin. That pile of coin is called grana. Pile of coin is called grana. And the thylakoid, two different types of thylakoids are there. Some of grana thylakoid, which is there in grana, and separately they have stroma thylakoid is also there. That means they are present singly, separately in the stroma. It can also be like that. This is the fret membrane which connects all the thylakoids. This is about a typical structure of plastids. This plastid or chloroplast, it is mainly involved in the process of photosynthesis. This is about the plastids. The next organelle we are going to talk about the sparosomes. Sparosomes, glyoxysomes, and the cytoskeletons, structures of cytoskeletons, and about we're going to talk about the nucleus, the structure of nucleus, and chromosomes and the chromosome structure. All these things in next class we'll do it. Okay, children, we'll do all the other things in next class. Next class, I'll just inform you, the next class is going to be on 19th. We are going to have the next class. Okay, children, if possible I can have one more class on Monday also, that I'll inform you later. Probably we'll have class on 16th also. I'll just inform you later, children. So now, as of now, 19th class is confirmed, and for Madesh, Madesh, you'll be having class tomorrow also, because you're going to attend both 11th and 12th standard simultaneously. So you'll be having class tomorrow also, biology class tomorrow. Tomorrow between 5 to 6:30 we'll be having a class. Okay, you can take my number and you can send a WhatsApp message. In that I'll store your number. I'll send the link by tomorrow by 4:45, and 5 o'clock you can join the class. So 5 to 6:30 you'll be having a biology class tomorrow. Where I want to discuss about human health and diseases. That is the topic. So you can attend the class tomorrow. Others, those who attended the test, just correct the paper and send it to me, all the answers by 9 o'clock, before 9 o'clock. And children, for all of you, happy Diwali. So this is my wishes to you. Have a safe and bright future. So let this Diwali brings more happiness to you, and also let it shine more brighter in your life, so that you'll have lot of knowledge and wisdom. And whatever you do in academics and all other things, I want you to shine. I want all of you to be like this, a brightest star or brightest lamp in your school, and perform all your tests, and whatever task is given to you, I want you to perform in a better way, so that you will become the best person in the world. Okay, children, thank you. Next, we asked the screen to be shared, right? Okay, 1 minute. You can copy this slide, children, for another 2 minutes. You can copy this slide. So etioplast is a precursor of chlorophyll. It helps in precursor of chlorophyll, and your doubt, Vashini. Centrioles are not present in plant cell. How does cell division occurs in plant cells? So here in cell division, mostly the cell division that occurs in plant cells, there are reproductive process. Got it? They are reproductive process. Whereas the growth is not much involved. The cell division is not much involved in growth. So there is no separate organelles are there. But for the cell division in plants, the chromosomes play an important role. The chromosomes are playing an important role, but there is no specified or specialized organ is there for cell division in plants. But the division, that is cytokinesis process and karyokinesis process, they are all directed by a structure called phragmoplast. Even the formation of the phragmoplast, it is done by the chromosomes. Okay, child. Thank you very much. If you have doubt, I'll clarify a doubt. Otherwise, we'll meet you the next class. I'll inform next class about the next class later. Probably you'll have class on Monday or on Thursday. Okay, children. Thank you very much. Be safe, and we'll meet you next week. Thank you, children.